Good morning. So this is our Wednesday morning session for the weekly live online sessions, and we're now on the week 5. So a couple of things that I wanted to have a look of this week, where we'll be focusing on the anatomical structure of the heart as a continuation of our review of the cardiovascular system. I wanted to just say a couple of words about the exam and lab practical, which we completed last week. And then again, kind of the same blurb you've seen before about the missing and assignments, retaking assignments, and all that. On this week's module, we'll focus, of course, on just quickly looking through what are the assignments, what are the due dates. And one of the things that you'll notice, we are completing this week labs, and there's 2 of those, and they're a little different from the labs that we've seen before. There's something that gets called Phil's labs. So we'll have a look of that. And of course, the main concept or main focus of our session for this week is going to be on reviewing the material. And for the heart, we'll look at the anatomy and physiology almost as separate blocks, although, of course, as you can certainly imagine, they are very much interlinked. And I just want to finish off with a little bit of reminders about the tutoring, one-on-one meetings, and make time for questions and answers. And if you are watching a recording, remember that you are not going to miss out on those questions and answers. You can always reach out to me, and I'll be more than happy to help with that. So just a really quick kind of update where we are with the exam 1 and lab practical 1. So I want to start by saying a massive thank you for everyone who completed the exam and practical last week. I know that might have been a little not so much nerve-wracking, but it's always a new situation when you're taking the first exam, first practical in a new course, and especially with a new instructor to you, since there is a little bit of variety in the format. And I approach the concept of testing in a little different, especially now on a time when we are working remotely, than the traditional testing where students have been traditionally required to just memorize stuff. My goal is that you actually go beyond that and you're able to integrate the information, you're able to apply the information, and so on. So that's why I'm not that concerned with making you just remember random facts. And as I'm sure you did notice, we did not use responders. So that goes back to that philosophy that I mentioned to you, that I think that the way the exams are set up and practicals are set up, that's the reason we don't have to use the responders. So what we do instead, we have a timeline, how much time you've got to take those per question, and we also try to have a variety of questions from one semester to another. One of the big things to notice here is that I will have to do the manual review of your answers, and I'm at large finished with that. I have a little bit more to do, and I typically don't release the final exam grades until everyone has completed. So there were few students who, for one reason or another, were not able to complete their exam 1 and lab practical 1 last week. I'm expecting that they're completed by the end of the day by large. We'll have a look of that. But I'm expecting for everyone to have your grades released by the end of this week. So you can stay tuned on that. And as I said before, I will send an announcement of that, mention that in the announcement when the grades are ready. So what you're seeing now is not your final grade, and it is very likely that the grade will actually go upwards as the spelling errors get checked on, so you won't lose points for anything like that. And of course, you had the extra credit question in the exam as well as the final question in there. Once I have finished the reviewing the exams and the final grades are released. I'm just putting this as a bug into your ear at this point, so you are aware that there is a plan in place for that. If you feel that there was questions that you kind of didn't understand, and even based on the feedback you don't understand, I really am super happy to do one-on-one get-togethers to discuss with you why the answer was what it was. I really strongly encourage you all to review the feedback that I give. I typically give quite a lot of feedback for the exams and practicals. And the big thing with feedback is that if you're getting a lot of feedback, it's usually on those questions where you missed out on something. So I try to explain on that written feedback why the answer was what it was, and what I can see from your thinking process, what might have left you stray from catching that answer. So I really encourage you to review that. Like I said, we're not quite yet there, but we'll see. Once the grades, final grades for those are released, I really would ask you to take the time to review the feedback. In terms of reattempting, if there is a valid reason why you did not do well, or if you totally bombed an exam or lab practical, I am open to discuss, have a look at, okay, where are we standing? Where does it place you? Should we consider that you reattempt an exam or a practical? But that's not automatic, so we'll have that discussion at that time. So just a bug of those things. So stay tuned, look for the end of the week when I should have your final grades for the first exam and lab practical one ready. I mentioned this before, that I am quite open for any missing assignments to be opened again for you to retake assignments if you feel that something didn't go the way you expected it to go. And I'm just kind of listed here a couple of questions that you might ask from yourself. You don't really need to explain them to me, but if you are ending to retake assignments, it's usually a good idea to think that, okay, I'm retaking an assignment now. What did not work last time? So I would first of all encourage you to review the feedback that you've gotten, whether it's automated or from me, that, okay, what were the answers that you were actually missing? Why didn't go to your way? Maybe there is something to do with the learning strategies or assignment completion strategies. Maybe we can work on that. I have a great amount of study tips, tricks, as advice on that. That's something that I used to do quite largely with my students and students from other classes. So if you feel that you would benefit from doing a bit of a review of what are good learning strategies, what are good exam taking strategies, what are good assignment completion strategies, just let me know. I might be able to just upload some resources to your Canvas course. And in addition to that, I'm super happy to take the time. We can sit down and figure out, how do we get you to succeed even better? The big thing is that review the previous assignment before taking it again beforehand. So how do you get to retake an assignment, or how do you get to have an access to missing assignment that's already past due? Big thing is that message me directly. I encourage the Canvas communication because the Canvas is the one that I normally answer the quickest, but I do, of course, also answer on the emails. But message me directly and include the details of what specific assignment are we talking about, because we do tend to have a lot of assignments, and if it's too vague, it takes me a little bit of time to figure out. I might need to reach out to you again to figure out that. Let's jump back. That what was the assignment that we're looking at. So just include what week, what assignment, and we should be good to have those open to you. So this week we've got 2 labs. And those 2 labs are a little different from what you've seen before. I was meaning to have a pen on, so apologies for that. So those 2 labs are a little different from what you've seen before. in a way of how the actually lab technically runs. However, what you are familiar with is the LearnSmart, which was basically just reviewing the material with the textbook. You have an access to link to the textbook for the particular part if you don't know a question. Also the quiz, which is like any traditional quiz, and the practice anatomy, which was mainly about preparing you for the practical. But you'll notice they're all, again, due at the end of the week on the Sunday, just 1 minute before the midnight. So I encourage you to make sure that those get done by that time so that you get the automated grades updated. And we'll discuss a little bit. What I want to take the time is that what is different about these labs from what you've done before. Of course, all of these are completed through your learning management system that's linked to the publisher's platform Connect. So they should be directly linked, and that's how we get your grades also to update directly on the Canvas. What I wanted to say was that I know that I promised you that we kind of hone down in the amount of homework, especially in terms of the LearnSmarts and quizzes. And I asked you to hold back a little bit from working on the week 2 material until the start of this week that started. These students were quicker than I was. So they started already the week before or a couple of weeks ago. So for that reason, I was not able to go back and adjust the amount of material. So those students beat me in terms of the timeline, but that does not mean that I've forgotten my promise. It just means that we were not able to cut down the amount of questions on these, but we'll do instead, I will cut down the amount of questions from the blood vessels chapter onwards, so from next week onwards. So if I may ask, please, 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 for the week 6, hold on to starting to work on those assignments until we actually get to the week 6. And then I'll make it so that it's all fair game. You can start working ahead. So I do realize that it might be a little bit of a bummer in terms of that the LearnSmart and quiz have quite a few questions this week as well as in the previous weeks. But that was just because of a couple of students having worked ahead of the time. So once they have been attempted those quizzes and LearnSmarts, I cannot change the amount of material for the rest of the class. So I apologize for that. I'm, of course, extremely excited and happy that we have students who were eager to make a head start. But if you can just wait until next week with the next week's material, we'll make sure that the amount of homework is gonna be manageable for you. So let's have a look of the labs. So the abnormal ECG and heart sounds are the 2 things that we're gonna be looking at from the chapter this week, the chapter on the heart. And these labs were different. And I know that the Flash player, the support for that ended at the end of last year. I believe that it was at the very last day. I might be off from the date, but so Flash player is no longer supported, and for that reason, all of the publishers are moving away from these. So I was expecting to actually update something different for these labs, but I see that few students were able to access these, like I said a little early, so they have been able to complete them regardless. So we're gonna leave them as they are. However, if you are not able to run these labs, maybe because of the Flash or any other reasons, I have an alternative lab for both that I'll be uploading very, very, very soon. So I'll give an update about what that alternative way of completing these is in just a few moments. I'll be posting an announcement about that. But if you're able to access these, great. So what we'll do, we'll go through the way how you were able to access these in the past. But again, if it doesn't work for you, don't think that it's your fault. It's probably because the Flash is no longer supported, and that for that reason, most browsers and most publishers have moved away from that. So you'll notice that I actually have a specific section that I built on the canvas, helping you to work out that how should you tackle these fill slabs, because it does involve you enabling the Flash player. And that's the key thing there. And enabling the Flash player to run these labs actually can involve enabling it in a couple of times. There is a chance that it just for some reason or another doesn't get fully accepted or opened up the Flash player for that assignment or that activity during the first choice. So it might be that if you get checked that a couple of times in the different places, depending on the browser that you use. And like I said, I fully understand that Flash Player is no longer supported, so I would not push you to have to use Flash Player because it doesn't have the regular safety updates. So if you are not able to run these labs, the alternative lab option is coming up really soon, and I'll send an announcement of that. But we'll have a look at how to access these if you are gonna try to access them with Flash. So, like we mentioned already, to work on these field labs, they are Flash based. So you have to have the Flash player installed, and that normally happens. That's not normally an issue. Let me just change the color there. I feel a little bit weird that the text doesn't show up. So what you need to do, you need to have the Flash player installed. So I assume that most, if you don't have a brand new computer or operating system, most of you would have the Flash player installed. That's not the big issue. But the bigger issue is that most of us, the Flash player is not enabled at all times. So you need to manually enable it to run. So basically, in other words, allow for it to be run. So you might see a screen that's something like what you have uploaded here on the left bottom corner, stating, for example, along the lines that you need to have an Adobe Flash player to view these labs. So get the Flash player installed. And that used to work, and that pretty much fixed the problem in the past. I don't even know if it is possible to install now Flash Player since it's no longer supported. So if you are able to do that, good for you. If you're not able to do that, we are gonna go to the alternative assignment. But if you have the Flash Player already installed, you might run into the fact that the browser automatically blocks it from running. Where you want to look to the very end of the address bar, regardless of what browser you use. Usually there is an address bar where you write the address of whatever your website you're going for. And at the end of that address bar, there's usually various different symbols. It might be a star for bookmarking some sites. But what you'll also might see is some sort of a symbol that looks like a lock, and typically a slash through that, or a circle and a slash. Something that shows that the lock has been blocked. What you want to do, you want to click on that symbol and choose Allow Flash to run. The other place where you might see, and it might be that you need to do both of these, is at this very other end of the address bar, there might be a symbol of the exclamation mark or some sort of another announcement. The exclamation mark, I believe, is the most common one that I've seen. And if you see that symbol, that's often another signal that the Flash player has been blocked. So click on that, and from the drop-down menu, choose to allow to run the Flash. So it might be that you need to do it on both of these, and only then the flash will run. It might be that you have to do it on only one. It depends. But if you allow the flash to run in every place that it might ask for that, what you'll end up seeing is a page that we'll have a look in a second. So here's a good example of that exclamation mark. And when you click on that, you'll see that you get a drop-down menu next to the simple flash where you can then choose that allow the flash to run. And if everything works out great, this is what you end up seeing. You end up seeing a menu with the fills lab. And this menu is an interactive kind of a table of index. And what you want to do from there, you want to choose the right assignment that you'll be working on. So I think that we had, in terms of the hard, abnormal ECG was one of the assignments. So that would be assignment number 30. And then we had hard sounds, I believe, as the second activity. And I'm trying to find it here. Well, you'll see it. My screen is a little bit small on the text. Oh, here it is. Hard sounds. I think it might be activity 26. It might not be, but I think it might be activity 26. Either way, whatever activity it is, you find the abnormal hard sounds and the abnormal ECG, or it might be normal hard sounds, and you click on that activity. Just choose the activity that matches with the name of the lab that I'm asking you to complete, and then you'll be able to work through that. So kind of having a step-by-step guide, you click begin, continue, click here to access fills, and this is the menu that you'll end up in. And on this menu, like we saw, the abnormal ECGs was chapter 30. You click next. You can do pre-lab if you want. You don't have to do the pre-lab. You complete the lab exercise, and you also complete the post-lab questions. The post-lab questions are actually completed in a previous window that is left open there in the Connect. So it's super important that you complete this post-lab to get the full marks for having completed this lab. Pre-lab was optional, but the lab exercise and post-lab, they need to be done. So you get the points. And if you get stuck, you can always contact the McGraw-Hill technical support. However, like I said, I realized that the Flash player is no longer supported, so it might be that they might not be able to guide you much further. So that's all that we need to know about the technicalities of this week's homework. What I will be focusing now on is the key points of the chapter material for this week. And I'll just take a very quick break and we'll continue focusing on what's important about the chapter material on this week's on the chapter of the heart. We'll do that in just a second. So let me just pause and I'll join you right away. So now we're back and we're gonna look at the key concepts of this chapter on the heart. So let's go through some of the important concepts that I want to highlight from the lecture material on this week's material. However, do remember that for the full story and full material, I always encourage you to refer the complete lecture videos that I have prepared for you and are available for you on YouTube via the links that I've uploaded to the Canvas. So let's have a look what we need to know about this chapter. I think that since we are talking about the cardiac muscle as the muscle that relates to the heart and structures of the heart, it's a good time for us to remind ourselves of the three different muscle types. So you remember we saw the skeletal muscles for the big muscles of the body, the muscles that we most commonly think about as the muscles that generate movement. Their microscopic structure, if we look at that, provided I get a pen up, here we have, is if we look at it a little bit diagrammatically, these cardiac muscle cells of these muscle cells of the skeletal muscle are cylinder in shape, and they are striated. So we can see those striations like zebra stripes throughout the entire length of the cell. And we also find multiple nuclei in these cells, and these nuclei are typically located on the edges of the cell. So that's our skeletal muscles. The second muscle type that you might remember from our previous conversations is going to be the smooth muscles, which we end up finding in the intestines and on the walls of the blood vessels. So these are muscles that control the flow of the material through the intestinal system and also control the blood pressure within the vessel. These muscle cells are kind of a spindle-shaped, if you wish, on their relaxed shape. We do not find any striations on those, and they have one central nucleus. When they constrict, the shape of the cell might be a little different, but at large, this is the shape of them. But where our focus will be on this week, since we are focusing on the heart and heart muscle, is of course going to be on the cardiac muscle. So let's have a look of the key characteristics of that and take it from there. So what we end up noticing there, that cardiac muscle is of course found only within the heart. And when we look at an individual cardiac muscle cell, they are typically these branched cells with a varying number of branches. I'm just doing one here. It looks, and this one, for example, a little bit like X-shaped with two branches on both ends of that cell. We do end up finding that they do have striations. So again, those zebra stripes, if you want to use that as a mnemonic, throughout the entire cell. They have typically one central nucleus. And the key characteristic that we end up finding is that at the very end of the cardiac muscle cells, we have these special adaptations of intercalated discs. So at the end of these heart muscle cells, we see these intercalated discs. These intercalated discs are structures that help with connecting one heart muscle cell to the next heart muscle cell. So they serve a purpose in that. So they facilitate the easy passage of materials and messages from one cell to another. And this is important in case of a heart muscle, especially because we want to have a synchronized contraction of all of the cells that form that tissue at one time. So these intercalated discs facilitate that. So that's characteristic for the cardiac muscle cells. So let's continue and see what else we might possibly want to highlight when we are discussing. So here we have a rather nice diagram highlighting this structure that we just talked about, the intercalated discs connecting one cardiac muscle cell to another cardiac muscle cell. But we'll also end up noticing that the cardiac muscle cells are rich in mitochondria. And if you think of back to the intro bio, basic bio classes, we remember that mitochondria were, of course, the structures that were responsible for the production of the ATP from the energy resources. So making the energy in a format that's available for the cells to use. So that's why we end up finding a lot of mitochondria in these cardiac muscle cells, that they need a lot of energy. They're constantly working. If you think, when does heart muscle stop working? Well, that's only really once we are dead. So heart muscle is constantly working. It starts to beat somewhere around 2 weeks of after fertilization, and it is really at the last breath, our last heartbeat, where it stops beating. So I did some math, and I kind of thought about that, since heart is never at rest. In a day, typical day, we pump about 7,000 liters of blood, and that results in somewhat 2.5 billion beats in a lifetime. Of course, we see that different animals have different cardiovascular systems. There might be different adaptations from what we see in a human, where we have the closed loop one-way system, but that's outside the scope of what we are gonna be covering on this course. I do think that comparative anatomy and physiology is always super useful, but we'll save that for later. And on that note, I think that we are ready to move on again. So let's have a look of the next section. So when we talk where is the heart located, heart is located just above the diaphragm. So remember to use the proper anatomical language. So that would be superior, so above of the diaphragm that I've symbolized here on this line. So that's the large sheet-like thin muscle. Expanding between the ribs 2 and 5, so the intercostal spaces, these spaces between the ribs 2 and 3, 3 and 4, and 4 and 5 are the ideal locations for listening the heart and heart sounds. If we look at the heart at large, heart is shaped a little bit like a cone, especially the ventricles. So you'll notice that we have 2 ventricles, left and right, and it is the left ventricle that has the thicker wall, more muscular wall, whereas the right ventricle, the wall is a little thinner in the thickness. That tells us that the blood that gets pumped out of this left ventricle needs a little bit more force for the pumping. Kind of jumping back to what I said using about the correct anatomical language, what else could we do to describe the location of the heart? It is posterior to the rib cage, so behind. It is anterior to the spine or vertebral column. Often we talk about what's the best way to someone's heart, and we hear that food or cooking, I'd say between the 2nd and 5th intercostal space. That's a little bit of anatomy humor for you. So what we're going to start with, we're going to look at the fact that heart is located inside a sac, really. And this sac plays an important role. So let's discuss that a little bit more. So heart is located inside what we describe as a pericardial sac. This pericardium surrounds the heart, and inside it, between the heart muscle and this pericardial layer, is our pericardial cavity. And within the pericardial cavity, we find a little bit of fluid that allows lubrication and movement of the heart. The reason why this pericardial sac is so important is that it allows the heart to move without being constricted by all these other structures that are enclosing or surrounding the heart so directly. So that's how we have this limit of the or we remove these limitations to the move. And what I want to do next, I want to talk a little bit about the heart wall structure. So here we see that if we work our way from the inside of the heart to the outside of the heart and look at this muscle wall that we're seeing here, the innermost layer is going to be endocardium. I always think of that like that is the most internal one. After that, the thickest layer of the heart muscle is going to be myocardium. That's the fleshy, meaty part of the heart muscle. And the outermost layer of the muscle, heart muscle, is going to be epicardium. Remember when we talk about geography, and this is not going to be a geography lesson. I just try to throw out various ways to remember things. When we have, here we have an earth. And when we have an earthquake, earthquakes rarely happen on the surface of the earth. They happen a little bit inside. So let's say this is not to the scale, but there would be an earthquake inside the earth there. This point on the top of the earth that's right above where the actual earthquake happened. That would be known as epicenter. So this epi refers to something that's on the top. So that's the outermost layer. So that might help you to remember the difference between epi and endo, and myo is then in between. So hopefully that helps to make sense about these heart layers. And let's look forwards. I want to talk a little bit about the clinical relevance of this. So I think it's always useful when we know the normal to look at a little bit of abnormal. So in this case, we can talk about pericarditis. And anyone who's done a medical terminology class already at this point will remember that whenever we have a term itis, the itis referred to an inflammation of something. So in this case, it's going to be inflammation of the pericardium. So what happens in this case? This causes a distinctive change in the sound that we will hear with the stethoscope, kind of this creaking sound as a result of the fluid leaking into the pericardial sac in a greater amount, and that will have an effect on the heart's pumping ability. So some of the treatment of interventional options might be trying to remove some of that fluid. But the big thing there is that, remember, we can hear a lot about the shape of the condition of the heart just simply with basic tools such as stethoscope. So what we're going to do next is that we're going to look at the two kinds of circulation that we have in the cardiovascular system. So sometimes what we describe about the human heart and human cardiovascular system is that it's almost like two hearts beating as one. That's kind of a romantic way of describing it. So what we have two circulatory systems, if you wish, or two loops for the blood to circulate in. And as we are starting to review through this, I want to take our attention back to what we covered in last chapter when we talked about the color of the blood. And we noticed that if it is highly rich, or blood that's highly rich in oxygen, so there's a great amount of oxygen in blood, that showed up as brighter red in color. So we talk about oxygenated blood. This blood typically comes from lungs where it has been re-oxygenated. So new oxygen has been added to the blood. In comparison, blood where we have a slow amount of oxygen, that's low in oxygen concentration, is referred as being deoxygenated. So that typically comes from the body and especially of the other parts of the systemic circuit of the body. And apologies for that. I have many windows open on top of what we're seeing here. So this is blood that's returning back to the heart from the tissues where the oxygen has been taken up and used. So remember this color difference as we are discussing of the flow of the blood through the circulatory system, but that also allows us to notice the flow of the blood within the heart. So what we'll end up noticing is that the heart can be roughly divided into half, and on our right hand side of the heart we have this oxygen low blood. So we have this deoxygenated blood. So this blood is coming from the body and it is low in oxygen concentration. That's our right side of the body. And remember that the right and left, we always talk of those as in relation to the patient's right and left. So this patient is lying in front of us on its back in this diagram if we try to position ourselves. So what seems to us as our left side is the patient's right side. So we use the patient's right and left terminology. Well, in terms of then on the left-hand side of the patient's heart, this blood is typically returning from the lungs where it has been reoxygenated. So this blood is rich in oxygen. We talk about oxygenated blood. So remember the difference between the two sides of the heart when we work through this material. So we're going to start with the first circuit, which is going to be the pulmonary circuit. And what we're seeing here is that the blood is coming from the right side of the heart. And this blood is low in oxygen concentration, so it's deoxygenated blood. It travels to the lungs, which we can see here, and it gets reoxygenated. So it receives now oxygen from the lungs and it returns as oxygen, rich oxygenated blood to the left side of the heart. So this circuit is known as pulmonary circuit, so referring to the lungs. The second circuit that we see is going to be the systemic circuit. So this doesn't cover the lungs, but it covers all other parts of the body. So on this circuit, the blood leaves the heart from our left side as oxygenated blood. It reaches tissues where to the cells it gives up the oxygen. So now it becomes low in oxygen concentration. So it's going to be our deoxygenated blood and returns to the heart to the right hand side as such. So that's the systemic circuit, which really covers all parts of the circulatory system with the exception of the lungs. So if we think about the size of these circulatory systems, this diagram, of course, is not to the scale, but it is there to give us a little bit of a clue of that. What we'll end up noticing is that the distance that the blood has to travel from the right-hand side to the lungs and then back to the heart, this distance is going to be much shorter. And there's going to be less of this capillary network area also to cover. So there is going to be a very low pressure on this circulatory system. We don't need a lot of force for the blood as it gets pumped out. In comparison, pumping blood to all other parts of the body from our left-hand side, and then it returning back to our right-hand side of the heart. That covers a large area, and in addition, it covers a large capillary network area as well. So this blood needs to be pumped to this systemic circuit with high pressure. We need a lot of force for that blood to do this journey. So that's really the big difference of why do we have this pressure difference between the two circulatory systems. So I prefer to use the full explanation to understand these differences. Don't just memorize the difference, but try to understand the reason why do we see this difference between the pulmonary and systemic circuits pressures. So low for the pulmonary circuit, high for the systemic circuit. Now that we kind of put the heart into its place, thinking about the whole picture of the pulmonary circuits, I think that brings us to a place where we can really start focusing on the different parts of the heart. And here we have a rather nice diagram of the heart. I prefer to use diagrams I learned visually. So what we'll do, we'll look at the 4 different chambers that make up the heart. And those 4 different chambers allow us then to fill in all the other blanks. We'll see various different kinds of valves. We'll see various different kinds of wall adaptations in the heart. But let's start with those 4 different chambers. And a good way to describe a heart is, if you want to make it simple for yourself, just think of it as 4 boxes piled on top of each other. And you might be asking me that how does that apply to what we just saw? So let's jump back to there. And what we see is that we really have these chambers that we notice with the empty hollow spaces inside the heart. And they form 4 boxes within the heart. Yes, the size of the boxes is slightly different than fully symmetrical, but we will just look past it. So always in A&P, try to simplify concepts, learn the simple version before you add more to that. So we see the 4 boxes that we will use to describe different chambers of the heart. And how we are going to tackle this is by giving them their names. So on the right-hand side, and remember this is the patient's right, we have right atrium and right ventricle. So right atrium is the more superior chamber on the right-hand side. And on the left-hand side, we have, again, left atrium and left ventricle. So ventricles are the more inferior chambers. So when we are looking at these chambers, what I tend to do is just abbreviate them. So we're marking the site, and we're marking whether we're talking about atriums, which were the more superior chambers, and ventricles, which were the more inferior chambers. So if we jump back to what we just saw here, and let's get a pen that's going to show up. We are going to see again our right atrium, right ventricle, left atrium, and left ventricle. Just as we saw here with these boxes. And we remember again, the patient's side is the one that defines the right and left, and atriums at the top, ventricles at the bottom. Well, let's have a look next. The flow of blood through these different chambers. Well, blood enters the heart on the right to the right atrium. That's where the blood comes first. And this blood is coming back from the systemic circuit. So it's oxygen-poor blood. So there's very little oxygen in this blood. It enters to the right atrium, and from there it passes to the right ventricle. This blood, still remember, is gonna be oxygen poor. From right ventricle, this blood flows out, and it actually doesn't go back to the right atrium, so we could think of this as a dotted line, but it just leaves and it travels to the lungs. And within the lungs, this blood, which was oxygen poor, it now gets reoxygenized, and so it's rich in oxygen as it returns from the lungs to the left atrium. And once this blood has collected as now oxygen rich coming back from the lungs, it moves to the left ventricle, from where it ends up leaving. And again, it doesn't go back to the left atrium, it kind of passes behind it and then travels and heads to the rest of the body to the systemic circuit. So serving all other tissues. So we can really see the same organization of the chambers and flow of the blood. The right atrium receiving blood from two blood vessels. These are gonna be something that we focus on the next week's chapter. Just if you wanna have complete story, the inferior and superior vena cava. So blood is oxygen poor, and now arrives to the right atrium. From there, it travels to the right ventricle, and from right ventricle leaves to the lungs. And now, being oxygen-rich blood, it returns from the lungs to the left atrium, from left atrium to the left ventricle, and out from the left ventricle to the systemic circuit, to the rest of the body as oxygen-rich blood. So we see the right atrium, left atrium, right ventricle, left ventricle, just as we discussed before with this diagram of the boxes. One structure that I do want to highlight, and I often ask from my students because it is a structure that becomes important later on as we discuss the surface anatomy of the heart, is going to be this wall that separates the two ventricles. This wall that separates the two ventricles is known as interventricular septum. So septum is always a wall that separates something, and if it's between the two ventricles, it's gonna be interventricular. Think of the term internet. It connects two computers to each other. Intercontinental flights connects the two continents to each other, and so on and so on. So interventricular, the name actually tells us where it is and what it is as a structure. So I tend to ask about that in the exam, so that's a good heads up of a structure to know. And now we are ready to look at various different kinds of valves that we end up noticing that these chambers are separated from each other with. So let's have a look of these different valves that separated these chambers from each other. And we're going to start by looking at the valve that separates the right atrium from the right ventricle. And this valve here is going to be called our right AV valve. And the term AV refers that it separates the atrium from the ventricle. So it's going to be our atrioventricular valve. So that's the term that we use for this first valve that separated them on the right-hand side. On the left-hand side, then equally, not surprisingly, we have left AV valve. Again, separating the atria from the ventricle, so it's gonna be AV valve. However, what makes these valves sometimes difficult in terms of the names is that we have multiple different names that we can use for a single valve. So this was the ones that we start with, but we can also see that they have alternative names that get used. For the right AV valve, which we saw here earlier on, another name that we see sometimes being used comes from the actual anatomy of this valve. And what we're seeing here on the left upper hand corner is looking at the heart from the top down. So we have a superior view and we've cut the blood vessels that leave the heart so we can actually see the valves. And we're going to be focusing on these two ones as we are talking about AV valves. So let's start with the right AV valve. So right AV valve is sometimes also called tricuspid valve. So it was this valve here. And that comes from the fact that we see 1, 2, 3 cusps. So leaflets that make up this valve. There's 3 of those. So we see those 3 there. So tri comes from the number of those. So tricuspid valve refers to the number of these leaflets that make it. So right AV valve is interchangeable with the tricuspid. On the left-hand side then, the left AV valve is often described as also with a term of bicuspid valve. That's because it has 1 and 2 leaflets, 2 cusps that make up this valve. Another term, the third term that we sometimes see for this valve here is gonna be mitral valve. So it can be any of these terms, they all mean the same thing for this left AV valve. So just be aware of that. All of them can be used interchangeably, a little bit depending on what aspect of that valve are we wanting to highlight. Are we wanting to highlight the fact that it separates the atria and ventricle? Are we wanting to highlight the anatomical difference between the cusp numbers, or are we wanting to highlight something else? So that's our valves that separate the atria and ventricle. There's going to be 2 more sets of valves. These are going to be much easier in terms of their nomenclature, in terms of their names. So the valve that separates our right ventricle from the pulmonary trunk. Pulmonary trunk is this thick blood vessel that comes out from the right ventricle and then divides into 2 and further divisions, carrying the blood to different 2 sides of the lungs. So pulmonary trunk and the valve that separates the right ventricle from the pulmonary trunk is known as pulmonary valve. That's not a surprising terminology, that makes sense, I would dare to say. So that's the valve that we see here. And on the other side, on our left side, the valve that separates the left ventricle from the aorta, which is this thick, large blood vessel leaving the heart, heading to the systemic circulation. That's gonna be our aortic valve. These are sometimes, both of these, described as being also semilunar valves. That's because their leaflets seem like half-moon shaped, if you wish. If we go back here, you can see they look a little bit like half-moon shaped. So semilunar valves are used interchangeably to refer to both pulmonary and aortic valves. Pulmonary valve separates the right ventricle from the pulmonary trunk, and aortic valve separates the left ventricle from the aorta. So here we have all of these valves included, and you remembered that the right AV valve could also be tricuspid valve, and the left AV valve then bicuspid, or it could be also mitral valve. The pulmonary and aortic valves, they could be also described as semilunar valves. So that's our valves that we need to know about the heart. So that was what I wanted to share with you about the valves of the heart. If we want to put them to this box diagram that we were using earlier on, we can then see our right AV valve here. Remember that could be also called our tricuspid valve, and we would see the valve that separates the right ventricle to the pulmonary trunk. That was, of course, our pulmonary valve, also known as our semilunar valve. The valve that separated the left atrium from the left ventricle, left AV valve, or also could be our bicuspid valve based on the number of cusps, or even mitral valve. And the valve that separated the left ventricle from the aorta, of course, our aortic valve or semilunar valve, depending on which term you prefer to use. So we can have issues with the heart valves again. So let's have a look at a little bit of the pathological side. Again, when we see something that works correctly, it sometimes adds up to that understanding to look at what if something doesn't work well. When we talk about incompetent valves, there is a backflow of the blood. So that does not close properly these valves. In terms of vulvar stenosis, these valves don't open up properly, so the valves remain always somewhat constricted. So the difference between the backflow and constricted opening, we can hear that on a stethoscope as a particular sounds that we hear. And of course, we can, if this situation is too bad, try to replace a valve. And there's many different approaches to that. We can use that fully mechanical valve, which would be usually something that we would do on older individuals, because those mechanical valves are going to be stuck to the same size. So we don't really want to put that into a very young patient whose heart still keeps growing, because that valve wouldn't then respond to that growth and grow with the heart. We can use animal valves. Pig heart valves are fairly common ones. And of course, we can take valves from other dead individuals, so cavitary valves as well. These two last ones are good because they do tend to grow as the individual's heart size grows as well. Next, we're going to have a look of the surface anatomy of the heart. And this is why I wanted to highlight earlier on that we needed to know certain structures of especially internal anatomy. We talked about the interventricular septum, because when we study the surface of the heart, these terms will help to guide us. So there's certain blood vessels on the top of the heart, on the surface exterior surface of the heart, that are going to be important. And I focus mainly on the arteries, but to really understand these arteries, we're going to start on this concept by talking about the surface anatomy before we lay these arteries onto these surface anatomy structures. So let's have a look of those. And here, we're going to start first with the structure known as coronary sulcus. Coronary sulcus can be also described as the atrioventricular groove. So that separates the atria and ventricles. So we have atrias at the superior part of the heart and ventricles at the inferior part of the heart. So this groove that separates the two is going to be known as our coronary sulcus or atrioventricular groove. The next one that we're going to have a look of is going to be the anterior interventricular sulcus. So you remember the interventricular septum inside the heart? Well, the groove that separates the two ventricles and is visible on the surface of the heart is going to be interventricular sulcus. And there's actually two of these grooves. The groove that we see on the anterior side is of course going to be the anterior interventricular sulcus. So we have our ventricles on both sides of that sulcus. That's kind of a depression on the surface of the heart. And we have similarly another interventricular sulcus and another depression separating the ventricles on the posterior side. So it would be behind here. We're not going to see from this view, but later on I will illustrate it to you. Just similarly to what we saw on the anterior side. So these are important landmarks. And why they are important landmarks is, like I said, these are where the coronary arteries are located. And before we jump into talking about coronary arteries, these three vessels, I have to admit that heart and especially these three blood vessels is what I built my previous career on when I just did cardiac research. So I focused my career on studying these three blood vessels. These three blood vessels, these three arteries on the surface of the heart are so important because heart muscle is like any other tissue in the body. It requires oxygen and it requires nutrients. So we need to be able to supply the heart muscle with the oxygen and with the nutrients. And how we do that? Through blood, providing blood there. So heart does not supply just the rest of the body. It also supplies itself. And these coronary arteries are actually the shortest circulatory system, shortest circulatory loop in the entire body. And this is a trick question that I often like to ask in an exam, and not just me, but many instructors. Later on the next week, we'll have a look of all these branches that arise from the arch of aorta. But what you'll notice that coronary arteries are the first blood vessels that arise after the aortic valve. So they're the first ones, and they are these two branches that we see coming out of here. And these branches can divide into further arteries. And what we'll notice is that the left ventricle is the one that receives most of the blood supply because that is the muscle that works the hardest in the heart. So let's have a look of those. So we're going to start with the left coronary artery. So we'll see our aorta here. An aortic valve would be here. So the first one on to the side of the left side of the heart, the first blood vessel coronary artery that arises is going to be our left coronary artery, often abbreviated as LCA in a medical language. This left coronary artery divides really quickly into two branches. On the anterior side and running in the anterior interventricular sulcus, we have anterior interventricular artery. You could abbreviate it as anterior interventricular artery, AIA, or something that's probably in medicine more common, LAD, left anterior descending artery. So that's the one here. The other branch that comes up from the left coronary artery is going to be circumflex artery, sometimes abbreviated as LCX, left circumflex artery. Circumflex artery goes to the posterior side of the heart and continues there extensively. But we only see a short branch of that on the anterior side, which is this view, of course. The second branch arising right after this aortic valve is going to be our right coronary artery. And our right coronary artery will run here in the coronary sulcus, separating the artery and ventricle on the right hand side, and it will have many different branches. We're not going to go to these various branches. We're just simply going to focus on this main vessel here. So that's going to be our right coronary artery, and I've labeled it here. We already had a good chance to discuss it. There is going to be also veins that drain the blood from the surface of the heart. I'll leave that for you to have an independent look of, and I really don't emphasize that much, but that's something that you can have an independently look of. It's pretty much a structure of blood vessels that drains everything that we saw before with the arteries of the heart. And now we are ready to look at the physiological aspects of the heart. So we're going to be spending a little more time on that. So in terms of the physiology of the heart, we're going to start by having a look of the heart rhythm and heartbeat. So just as we had action potentials in the nervous system, we talked about them at the end of the BIO201, and an action potential looks something like this. If you remember the diagram that we did, there might have been a little bit more undershoot recovery period there. Well, what we'll end up noticing is that we have similar electronic currents in the heart as well. But the shape of these electric currents is going to be different slightly, and there's also going to be difference in the duration of these electrical currents. So let's have a look at that. So whereas the nerve cell action potential lasted somewhere between 2 to 5 milliseconds, the cardiac action potential is going to be in hundreds of milliseconds, typically between 2 to 400 milliseconds. So it is going to be much longer. And this length of the action potential in the cardiac cell and cardiac conduction system is going to be important, because it being longer gives enough time for these ventricles to fully contract and for the blood to move from one chamber of the heart to another and then to other parts of the circulatory system. So for that reason, we do need a much longer relay time or delay time as these action potentials progress through the heart. So that's something to keep in mind. All of these events I have listed here for you to have a look independently, or if you wish, I discuss them in a great detail during our full lecture video. What we see is that when we overlay the conduction of the action potentials in the heart, there is a specific order of events and specific passage of these. So let's go through that and have a look at it. So everything starts here And this structure that we see here is gonna be our SA node. So our sinoatrial node. Let's mark it there. So you could say that that's really our cardiac pacemaker. That's where the heart rhythm is controlled from. This generates the impulse that then spreads throughout the atrial walls, both on the right side and on the left side at the atria. And you'll notice that it spreads through these walls onto the both sides until it reaches this point here. And we'll focus on this point a little bit. This point that we see here is gonna be our AV node. AV, since it's located between the atria and ventricle of the right side. And this AV node works as a delay or relay station. So the propagation of this impulse through the various structures of the heart is delayed for a moment there. And after this momentarily delay, which allows for the atria to fully contract, this electrical current continues its conduction down to the bundle branches and then down to the bundle of his Purkinje fibers and eventually up to the ventricular wall. So now ventricles will contract, and at this point the atria have fully relaxed. So we always have this rhythm of the atria contracting, ventricles being relaxed, and then ventricles contracting, atria being relaxed. So it's this rhythm where blood has always, in terms of the chambers, a place to go. And this rhythmic contraction was so important where we saw all these intercalated discs connecting these cardiac muscle cells, and we also see this slight delay of the propagation of the cardiac action potential. Well, sometimes we can have issues with the conduction of these electrical currents, and we can have issues with the heart rhythm. So you can end up with something known as an arrhythmia. Arrhythmia is when we have irregular heart rhythm. So what we would normally see as a regular pattern fails to take place. So we have irregularity there. This results in uncoordinated atrial and ventricular contractions. The other thing that can happen sometimes that these contractions become completely irregular, very rapid. Heart goes into fibrillation. And what we can do in these is to use this tool that many of you, I'm sure, have seen. Automatic external defibrillation. So what that does, that's an automated machine that you will simply follow the instructions that it gives you, and it will give a hard electric shock that aims to restabilize these heart rhythm contractions or these electrical currents within the heart so that heart returns to a normal pattern. Really, I would say that CPR is a good skill to know, but it is the AEDs that really save lives. So I really encourage you all to get familiar if you have a chance to how to use AEDs, how do they function, what do you can accomplish with those. Sometimes some patients might have regularly issues with these electrical problems within the heart, and they might have actually kind of an AED implanted within their body. These are pacemakers. So these pacemakers are small electronic devices that help to reestablish the heart electrical current contraction that control the contractions in patients who have regularly issues with those. Interesting part, as a side note, we can actually reuse those. So we do remove those from dead catheters, and they do get taken care of, reset, and can be reused. So that is possible. So that leads us to having a look of these electrical currents within the heart. And this is a technique that we call ECG, or sometimes as EKG. So electrocardiogram is a way to collect information about these electrical pulses, electrical currents within the heart. But what we're seeing is a summation of all various currents from various places of around the chest cavity. So but we don't see a single part of the heart and single parts electrical currents. We see the summation of all of the heart from that location where the lead that measures these is placed. So it is worthwhile to have a basic understanding of what electrocardiogram tells us. This wiggly line tells us a lot of information, just as we saw with the stethoscope that we could hear a lot of information. So let's have a look of a very stylized, very clarified trace of an ECG. And we see here about 1 second's worth of electrical currents. Different parts of this trace have specific names because they tell about specific events in the heart. First of all, this first bump that we see initially before these other events is gonna be our B wave. And I'm just looking, do I have it discussed somewhere differently? I think that this is a good time to discuss it. So we're gonna focus on it at now. So this B wave, it becomes visible first and is followed by a series of first a depression, then an upshoot, and then another higher depression. And these peaks on this series of events have each their own letter symbolizing them, Q, R, and S. So we often discuss this as a QRS complex, signifying all of these peaks. And the final larger bump that we see at the end is going to be our T wave. And why these are useful, that they tell about particular points of the cardiac cycle where these electrical currents tell us that is happening now. But we can also measure distances between these events to establish is the speed of these taking place in a way that we expect them to. So we're looking whether we see something and are the distances between these correct. So we can see many different kinds of intervals and segments between these. So when you're analyzing an ECG, those would be important things to look at. So we're going to have a look of now mechanical events. So we've discussed a lot about electrical events, but let's have a look of the mechanical events that take place within the heart. So we're going to have a look of these mechanical events as the next one. And a couple of terms that I want to discuss as this get us started with are the systole and diastole. During systole, there is a contraction of either the entire heart or the particular part that we're talking about. And during diastole, there's a relaxation of that part. How you can think of is that during systole, this part is contracted, made as small as possible, whereas during diastole, that part dilates, enlarges as large as possible. And both systoles and diastoles are part of the cardiac cycle, so one complete cycle of events, these heart events, is known as a cardiac cycle. This brings us to talk about the heart sound. So we hear certain particular sounds, usually sounding like lup-dup, 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 when we listen hard with a stethoscope. these sounds are a result of the heart valves closing. So the first sound, kind of, if you wish, this lub sound, is going to be about the closing of the AV valves, whereas the second, this dub sound, is then related to the closing of the semilunar valves, so our pulmonary and aortic valve. Between these, there is a pause, and this pause is the time when the heart is relaxed. So when we take a stethoscope, we can place a stethoscope into particular locations to focus on hearing particular parts of the heart. So depending on where you place the stethoscope, you can direct your main focus on a particular heart valve and listen how that seems to be working. So again, I think rather amazing how much information we can collect about the anatomy and physiology of the heart without even having invasively go in there, just from these external measures. If we hear something known as heart murmur, these are again abnormal heart sounds, which gives us an indication that there's some kind of an obstruction taking place. So this can be either incompetent valve, so valve that does fail to close, or stenotic valve, where the valve fails to open completely. So again, you can get trained on identifying these different sounds and having a better understanding of that. As we are getting ready to wrap up, I just want to show that we can also combine various different measures to have a complete understanding of what's actually happening. So cardiac output, CO, tells us the amount of the blood put out within a particular time period. Usually how much blood is pumped out in a minute. And cardiac output is a result of the summation of the heart rate. So how often, how many beats per minute we have, how often the heart is contracting, and how much blood is squashed out from the heart with each of these contractions. When you multiply these by each other, that gives us the heart rate, cardiac output, how much blood is pumped out of the heart within a single minute. Usually this is somewhere around, I would say, 5.25 liters per minute. So that's about 1.5 gallons, roughly. My imperial scale is not that strong, but it's quite a lot, I would say. And that wraps up everything that I had highlighted for you from this chapter. And I just want to wrap up by reminding you that we do have tutoring available for you. So this tutoring is available in an electronic format at the moment through the Cranium Cafe. You've seen this link in various places. No appointments required. You can pick the tutor that you like. If they're available, they will work immediately with you. If not, you'll just wait for them to become available at the lobby. In addition, I am available to discuss with you, have one-on-one sessions, have group sessions, or entire class sessions. You just let me know and we'll make that happen. We'll find a time that works best for you. As always, this would be the time when I would entertain any questions that you might have. But do remember that you can always also message me here on the Canvas, or in other ways. I aim to respond within 24 hours. So we'll get any questions, any concerns that you have addressed. I am here to help you. My goal is to make sure that you can succeed and you get the best possible grade that you can get on this course. And on that note, I do think that we have really covered everything that I set out for us to do during this session. So I very much thank you for joining me, and I look forward to a great week with you. Stay tuned for the further information that I have about these field labs. I know that they can be challenging, especially now that the Flash is no longer available. So I will update about those on our announcements in a due course. I hope you have a very good week. Remember, I'm here to assist you, and thank you for joining me today on this week 5 review session on art.